Okay. So, Min, you just want me to start?
I will start the introduction first,
Okay.
and then I'll introduce you. Okay, let's get started. So hello everyone. So welcome to the second week of the special lecture series. Again, my name is Min Zhang. I'm Professor of Statistics and the Associate Director of Data Science at Purdue University Center for Cancer Research. So this seminar series is part of our NCI-funded R25 summer workshop on big data training for cancer research. So today we have three panelists, John Fry, Nadia Lama, and myself. So John has helped with setting up the Zoom meeting and handling all the registrations. So he will also record the lecture and post the recording at our workshop website later. So now I would like to introduce our webinar moderator, Dr. Nadia Lama. Dr. Lama is a research assistant professor in the Department of Comparative Pathobiology at Purdue. So she has been the manager of the Purdue site of the Collaborative Core for Cancer Bioinformatics since 2015. She has a background in biochemistry, molecular biology, and computational biology. So Dr. Lama got her PhD in 2015 at Purdue, and her research interests include single-cell data analysis and data integration with a focus on immunological diseases of the prostate. So with that, I will hand it over to Nadia.
Thank you, Min. So welcome everyone. And just as per usual, we will keep you muted for the duration of the seminar, but please feel free to use the Q&A function at the bottom of the screen to ask questions throughout the seminar. And if we need clarification, John could unmute you later, but we will use this Q&A function to take care of the questions. And feel free to upvote questions that you are particularly interested in, and those will float to the top and have a greater likelihood of being answered. So now I'll hand it back to Min.
Thanks, Nadia. So it's my great pleasure today to introduce Dr. Hongzhe Li. So Dr. Li is a Perelman Professor of Biostatistics, Epidemiology, and Informatics at the Perelman School of Medicine at the University of Pennsylvania. So he's the Vice Chair of Research Integration, Director of the Center of Statistics in Big Data, and the former Chair of the Graduate Program in Biostat at Penn. Dr. Li has been elected as a Fellow of the American Statistical Association, a Fellow of the Institute of Mathematical Statistics, and a Fellow of AAAS. So Dr. Li served on the board of scientific counselors of the National Cancer Institute of NIH and regularly serves on various NIH study sections. He served as the chair of the section on statistics in genomics and genetics of the ASA. He's currently an associate editor of Biometrics, Statistica Sinica, and also the co-editor in chief of Statistics in Biosciences. So Dr. Li's research focuses on developing statistical and computational methods for analysis of large-scale genetic, genomics, and metagenomics data, and theory on high-dimensional statistics. So he has published papers in high-ranked journals including Science, Nature, Nature Genetics, Nature Methods, Nature Biomicrobiology, Science Translational Medicine, Cell Host, and Microbe, and also the stat journals like JASA, GRSB, Biometrica, and Annals of Statistics. So with that, so please join me in welcoming Dr. Li.
Great. Min, thank you very much for the kind introduction. You hear me okay, everybody? Great. So yeah, so today I'd like to share with you some of the recent work in the area of microbiome data analysis. Mainly, I'm going to briefly talk about 3 problems. One is related to how to incorporate the phylogenetic tree information in analyzing the microbiome data. And the second topic is on estimating the bacteria growth rate based on shotgun metagenomic sequencing. And the third topic is related to how to predict the biosynthetic gene clusters in the microbial genomes. So we know in human gut, there are billions of microorganisms, and altogether those microbes form the human gut microbiome. And human gut microbiome plays a very important role in human health and disease, including protection against pathogens, synthesis of proteins, and they are also related to immune system development, and also synthesis of the various small molecules and the metabolites. So microbiome is related to human immune system development and immunology. Here is an example. For example, the microbiome can metabolize the nutrient we obtain from the food we eat. For example, they can metabolize the carbohydrate or proteins into various type of small molecules, secondary metabolites, and metabolites. And some of the metabolites really can affect the host through activating some of the important genes, particularly for some of the host immune-related genes. So one of the questions we are actually really interested in understanding how microbiome can produce different metabolites under different biological conditions. And microbiome is also associated with the risk of cancer initiation and the progression of the cancer through different possible mechanisms. For example, some of the bacteria, such as H. pylori and F. nucleatum, those bacteria can directly lead to genomic instability. In addition, virus, for example, certain virus such as HPV can activate the oncogenes and the inflammation, and that can also lead to cancer. The third mechanism is if there is this barrier breach, is leaking of the bacteria into human host, and this can trigger the pro-inflammatory signals, and that also can potentially lead to cancer. The fourth mechanism would be so-called dysbiosis, means the shift of microbial community from the normal state can lead to immune dysregulation, and that can also lead to cancer. So in this field, in the cancer research field, people are really interested in developing microbiome-based individual treatment assignments. The basic idea is whether I can assign different treatment based on the composition of the microbial community. So I'd like to give you one example. This example is taken from a paper that was published in 2018 in Science, where the author in this paper, they look at the melanoma patients, their response to a very effective immunotherapy called anti-PD-1 immunotherapy. And what you see here, there are 2 plots from their paper. You can see here, the first plot is a survival curve for the melanoma patients stratified by the diversity of their gut microbiome. And you can see here for the group with a high microbial diversity, this is the blue line, you can see they have a longer survival time. So the progression-free survival time is much longer than the patients with a low or intermediate level of gut microbiome diversity. This is just look at the overall microbial community diversity, you can see different patients, they responded to the same treatment very differently. And similarly, on this plot F, what you see here are the plot of so-called coordination plot. Just think this as a two-dimensional dimension reduction of microbial community based on a certain microbial distance, microbiome distance. And here you can also see that the patients who responded to the anti-PD-1 immunotherapy and the patient who didn't respond, you can see they have a different overall microbial composition. So this is actually one of the really important publications in cancer research in linking the gut microbiome with response to the cancer treatment. So how are we going to study the gut microbiome? So now in the last, say 15 some years, because of this sequencing technology, which really allows us to perform shotgun metagenomic sequencing, which provide a way of studying our gut microbiome using the DNA footprint of those microorganisms. So the basic idea is the following. For example, if I want to study my gut microbiome, I can collect biosamples, for example, fecal samples. From the sample, I can extract the DNA and prepare the sequencing. You can do the shotgun metagenomic sequencing of all the DNAs you extracted from your sample. And eventually, you're going to obtain a very large data set, a tetrabyte of text file, which are the very short sequencing reads. And each read is about 100, not 200 base pair long. And for the many studies, for one single sample, you have tens of millions of reads. And usually you have hundreds of samples. So the original, the size of the original data set usually is very, very large. So people have been developing tools and computational method to try to understand the microbial community based on those shotgun metagenomic sequencing. For example, from this raw sequencing data, you can perform taxonomic profiling, try to understand what are the microorganisms in my gut microbial community. So you can profile all the bacteria, you can even profile the virus and other type of microorganisms. In addition, because those are the sequencing data sampled from the genome, different part of the genome, you can also try to understand the genes carried by those microorganisms. So you can also obtain the functional profile based on this data. And nowadays, people even study the genetic polymorphisms, look at the SNPs or the structure variation of the microbial genomes based on shotgun metagenomic sequencing data. So there are a lot of computational challenges and computational problems related to analyzing such data. So later on in my talk, I'm going to present another features that we think is also relevant to study microbial communities, which are the bacteria rate of replication, how fast those microorganisms are replicating in the community based on shotgun metagenomic sequencing data. So on the top of the taxonomic profiling, functional profiling, this data also provide useful information for understanding the growth dynamic of the microorganisms. So microbiome, the data from microbiome studies, the problem is indeed a big data problem. As I mentioned, for a typical sample, one single sample, usually we have tens of millions of very short sequencing reads. And each read is about 300 letter, letter long. And for a given sample, usually those reads are derived from hundreds of bacterial species mixed together, hundreds of different type of bacterial species, but they have many different abundance also. So, and in addition, in order to understand those millions of sequencing reads, we also need to have access to the reference database. For example, the current NCBI database include about 300,000 of bacteria, altogether bacterial genomes. And for typical genomes, we have about 5 million letter long for the typical microbial genomes. So it is a very big data size-wise. And so in my talk, I'm going to present 3 examples just to demonstrate the role of statistics in microbiome data analysis. So the first problem is related to analyzing the data on the phylogenetic tree. So what is a phylogenetic tree? Remember, we are studying many bacterial species, right? Not just a few, many bacterial species. And many of those species are evolutionary related. And phylogenetic tree give us a good summary of how they are related based on some of the universal marker genes or a set of universal marker genes. You can construct this type of phylogenetic tree. So in this tree, the branch usually the length of the branch represent the evolutionary distance. And each of the nodes, each of the edges, each of the nodes represent one bacterial species. So now I have my shotgun metagenomic sequencing data. I can place my sequencing reads to the branches of this phylogenetic tree. So the idea is everybody, we're going to have the same phylogenetic tree of the bacterial species, but my reads, my millions, I'm going to place my millions of reads to the branches of the tree. That's my data based on, so how do you place the reads to the branch of the tree? Basically, there are certain algorithm you can see, seems like your sequencing reads are derived some part of the phylogenetic tree. And your data, you have your sequencing reads, so you can place your sequencing reads to the same tree. So the difference between your data, your microbial community, and my microbial community can be summarized as the distribution of the reads on the branches of the tree, which we call the probability mass on the tree. Same structure, but we have different mass, different width of the, just think about probability mass as the width of those branches. Those are the difference. So in the last 2 years with my postdoc, Sule Wang, we made some progress in understanding how to analyze this type of data. So I just give you some summary about what we have done. For example, as I mentioned, you have your probability mass distribution. You have a true probability distribution of the reads on the tree, and I have my distribution of the sequencing reads on the tree. So just think about if there are 2 true probability mass distribution denoted by P and Q. Then one way to define the distance between your microbial community and my microbial community, taking into account the structure of the tree, is to calculate the distance, the so-called Wasserstein distance between the 2 microbial community. As it turned out, this because of the special tree structure, the Wasserstein distance between 2 microbial community or between 2 probability mass distribution can be very easily written as this closed form formula, which is sum over each of the branches. So E represents an edge of the tree over the set of edge of the tree. LE represents the width, the length, not the width, the length of the phylogenetic tree. Remember I said the length represents the evolutionary distance among the bacterial species. And the absolute value of the difference of the probability mass distribution in this edge E, in this not edge E, in the subtree below the edge E. So PE represent all the reads that are assigned below the edge E of this phylogenetic tree. And this is called the Wasserstein distance between 2 probability mass distribution. So those are the quantity that we really want to estimate. Right? We don't observe the true probability mass distributions, but what we observe are those sequencing reads, which can be interpreted as an empirical estimate of those probability mass. Right? So simple idea would be, I'm just going to replace the PE, the probability mass under the subtree of the branch E, by my empirical distribution of the distribution of the sequencing reads. And you just plug this in. This is natural estimate of this true Wasserstein distance. However, if you look carefully, this guy actually does not give a good estimate of the true distance. The reason is, so typically the distance is written as a sum of absolute value of the difference. So in theory, the plugin estimate may not be a good estimate when PE and the QE, the true values, are really close to each other. If they're really close to each other, means the difference is close to 0. Then absolute value, just think about the absolute, you want to estimate the absolute value of something close to 0. The empirical plugin estimate provide a biased estimate. Sometimes the bias can be really large if for many E, those true PE is the same as QE. So that's the basic idea. We want to improve the estimate of the water city distance. Also in the microbiome data analysis field, this distance is called the weighted Unifrac distance, which is extremely widely used distance in the literature. But we can improve this. So basic idea is we can, when the PE and QE are close to each other, we can develop a better polynomial approximation for the absolute value of that distance. And so use that better polynomial approximation for that difference, we can reduce the bias of the estimate of the distance. So that's the basic ideas. And this algorithm actually, because of this special tree structure of the phylogenetic tree, the algorithm can be run very fast. So I'm not going to go into details of our algorithm, but I want to just to demonstrate this improvement by using some simulated data and the real data set. So here is what we did. So what you see here, we can simulate the healthy microbial community basically by resampling the data from healthy individuals and create a 200 microbial community for 200 healthy individuals. We also created a 200 disease sample to just allow their community deviate from the healthy community. And in the field, what people often do is based on the healthy individual, you can compute the centroid, means the centroid of the composition of the bacteria, or the centroid of the probability mass distribution on the tree. Then for each of the individuals, you can compute the distance to the centroid. And you use that distance as a way to quantify the dysbiosis of the microbial community. Okay, so I have 200 healthy individuals. I have their probability mass distribution on the phylogenetic tree. Then I compute the centroid of that 200 distribution based on the healthy individuals. Then I'm going to compute the distance, which I just mentioned, the water stand distance to the centroid. So what you see here, the first plot on the left-hand side are the histogram of the distance to the centroid for the healthy individuals and for the diseased individuals. The first one is based on the plug-in estimate. You can see there's some shift in the distribution of the distance to the centroid, but the shift is not as large as our improved estimate. So on the right-hand side are the 200 distance to the centroid for the healthy individuals, and the 200 distance to the centroid for 200 disease samples. You can see a better separation based on our improved estimate of the distance. Okay, any questions here? Okay, so this basically said by apply our improved estimate, we can see a better separation of the microbial communities between the disease samples and the healthy samples. So now if you look at a small data set that we analyzed. So here we look at this is not a gut microbiome. This is actually the microbiome from the patient's colon biopsies. And here I have 16 Crohn's disease patients, but they are not in the active state. So they are not active Crohn's disease patients. and I also have data from 18 healthy controls. So we can apply our method to calculate all the pairwise distance. So all pairwise distance, and we can perform a distance-based test of the difference of the distribution of the microbial community. Okay, so you can perform certain tests. The test we use is a graph-based two-sample test for the overall distribution. And what you see here are the graph, and you can see here, those guys, most of the people in the center of this graph are the healthy, normal, healthy individuals. And in terms of the pairwise distance of the healthy individuals, you can see they are similar. It means the healthy, 18 healthy individuals, their microbial composition are similar. But for the patients with the Crohn's disease, their distance are further from the healthy individual. That's one fact we know. But another fact is they are not close to each other. So the patients, they deviate from the normal healthy microbial community, but they are not like, they are very similar, right? They are different in their unique way. In this sense, actually, I think there's a distance-based analysis truly makes sense, right? So normal individuals are similar, but each patient, they have their unique way of deviating from the healthy normal community. So if you use our proposed improved estimate of the distance and apply this graph-based two-sample test, we can see a p-value of 0.0099. If you just use a standard plug-in estimate, you have a slightly less significant result with p-value of 0.025. But the sample size is small enough. You don't see a dramatic difference, just slightly more significant result from our proposed estimate of the distance. Okay, so this is usually what people do. Once you have the data, you perform a global test to see whether there is indeed a difference between the healthy microbial communities and the disease microbial communities. But after you perform the global test, you also want to find out if there are any part of the phylogenetic tree that contribute to that difference. So basically, not only you want to see there's a difference, you want to see which part of this phylogenetic tree that shows the biggest difference in terms of the probability mass. In other words, in terms of the species or microbial taxon relative abundance between two different groups. So this is the next question is, where is the difference, how to identify the difference, okay? So this is, again, I mentioned this, for two microbial communities, we can just interpret their data as an empirical estimate of the probability mass distribution on the tree. And I mentioned this a phylogenetic distance, this is a Wasserstein distance. And this distance can be extended to a so-called weighted alpha distance. So Wasserstein distance basically is a distance where alpha being 1. So if alpha equals 1, this gives us the Wasserstein distance. But in the literature, people also take alpha equals 2, for example. So that's almost like a L2 norm distance. So based on the distance. So another fact I want to point out is this distance. See this distance is really can be written as a sum over each of the edges, and the difference of the probability mass of the subtree below a given edge. And this is what we call the optimal flow. So basically you look at the P probability distribution on the tree and the Q probability distribution of the tree. You take a branch, you look at all the probability mass below this edge E, you compute the difference. This is called the flow. So if you want to test two populations, for example, disease population and normal population, whether there's a difference in the population probability mass distribution, and this is equivalent to testing whether this vector of the flow, so this is a long vector, right? Because this is a vector with a length of number of the edges, whether this vector is 0. If this vector is 0, then the distance is 0. So how to test this null hypothesis of the optimal flow being 0, the whole vector being 0? And in literature, again, people often perform something called Permanova. It's permutation-based analysis of variance based on distance. So once you specify a pairwise distance, you can compute a Permanova statistics. In fact, the Permanova is equivalent to the L2 norm distance of the estimated population mass. So basically you have sample, IAD sample in each of the two populations, and you just take an average of the probability mass. This is my P hat. There's a bar. This bar means the average of the first population, the probability mass, at a subtree below the edge E, minus P2, the second population, the average of probability mass. This is just a Permanova, equivalent to Permanova. And based on this definition of Permanova, you can actually study some of the properties, such as the asymptotic distributions, such as the power, what determines the power of this test statistics. You can study that. So you can see the Permanova test statistic is really the sum type of statistics, right? You sum over all the edges of the difference of probability, empirical probability mass, and you take the square. So this is the best way to do the test or not. But that's going to depend on what is the alternative. See here. So now is this whole optimal flow being 0. Alternative is this vector is non-zero vector. Non-zero vector is two possible alternatives. One is every element of this delta vector is 0. Another one is only a few of them are 0. So depending on different possible null, you may be able to develop different tests, that are powerful. Okay, so Permanova is this a sum of the square type of the test. So, but if you look at the real data, this is the data I mentioned, the Crohn's disease in normal individuals. What you see here, x-axis are those those edges of the phylogenetic tree. The y-axis are the flow, are the average of the probability mass for the normal individual, between normal individual and the individual with Crohn's disease. And you can see most of them, they are pretty small. They are really close to zeros, except for some of the branches, you have you have a larger difference. So for this type of data, instead of your statistic, develop a statistics with a sum of the square of the difference, a better statistics would be you take the maximum of the absolute difference. So you take instead of the sum of the square, you take the maximum over all the edges of the normalized difference. You normalize by the variance. And this is what we call the max type of statistics as an alternative to Permanova. And this actually can be more powerful than the standard Permanova if the alternative is sparse. And I'll just show you one example. So back to this UC and Crohn's disease patients. So again, this is a very small study where we have 18, 14, 15 samples in each of the 3 groups. Those 3 groups are the first, the group A are the 18 healthy individuals. This is again is a colon microbiome, and the group B and C are the patients with UC, patients with UC, and treatment means B. So group B are the patients without treatment. C are the patients under treatment, and I just want to know whether there's a difference in the overall microbial composition, and if there's a difference, where is the difference on this phylogenetic tree. So here are the results. For example, every pair compare group A and B, A and C, and B and C. If you just do the standard Permanova type of test, you don't see anything significant here. There's no significant. But if you use our proposed statistics, which is a max type of statistics, actually you see a quite significant difference in the microbial composition between the group A and B. So between the healthy individuals and the UC patients without treatment. But if you have UC patients, if they were under treatment, that's between A and C, those two groups, you don't see a big difference. So basically, sometimes if the difference is only concentrated at a subset of the phylogenetic tree, by using this max type of statistics, you can gain power. And in addition, you can figure out roughly which part of the tree, which E lead to the maximum of that difference. And you can point it out on this phylogenetic tree, which part of the tree that show a difference. Okay, so this is something that we can do. Okay, so this is the first topic I want to talk about. Just to summarize this is, it's really important to account for the phylogenetic relationship among the bacterial species. And one way to do this is to incorporate the phylogenetic tree information into statistical test and into calculating the distance. Okay. So now I move on to the second example, which is related to estimating the bacterial growth rate based on shotgun metagenomic sequencing data. So here's the idea. Remember the data we only have a cross-sectional sample of the microbial communities. So we don't have any longitudinal observations. But the trick is, even that, you can still roughly get an estimate of how fast the bacteria are divided. So the idea is this: if you look at a given microbial communities, there are many, as I mentioned, billions of microorganisms. And some of the bacteria, for example, if they are actively dividing, if they are fast-growing bacterial population, you tend to see more new DNAs get replicated in the regions that are close to the replication origin. So you look at microbial genome, usually most of them you have this circular genomes. And so this group, I think the red color, are the bacteria that are actually divided, because you see more DNAs in the replication origins. But if you look at another color, green color, I think it's a green color, that group, you don't see many new DNAs got produced. So that means that particular bacteria population, they are not dividing very fast. And you have some bacteria that are in between. Okay, so those are the true microbial communities. But the data we have, remember, are the shotgun metagenomic sequencing data, are the reads that are sampled from those genomes. So the idea is, if you can align those sequencing reads back to the bacterial genomes, for the bacteria that are fast-growing bacteria, you're going to see this uneven reads coverage, right? So in the replication origin, you're going to see more sequencing reads that can be assigned to this origin as compared to the terminus region, right? So after you align those sequencing reads to the reference genome, hopefully you can get a plot like this to represent the uneven coverage of the reads. Then from which you can quantify the growth rate just by look at the slope or look at this peak to trough ratio. And this has to be done by a paper published in 2015. And this was the first paper that proposed this idea of studying bacterial growth rate based on uneven coverage of the sequencing reads relative to the genome, to the complete genome of the bacteria. But this method only works for the bacteria with known complete genomes. But if you analyze, if you have any experience with analyzing shotgun metagenomic sequencing data, is the fact that usually you have 40 to 50, sometimes 60% of the sequencing reads that cannot be aligned to the known bacterial species. That means there must be many other new bacteria that we don't have the complete genome sequence. But now the computational tool is powerful enough to allow us to perform metagenomic sequence assembly. So you can assemble the sequence. You can assemble the genome of potentially new bacteria species. And in literature, this is called a metagenome-assembled genome. There's a MAX. So here's the basic idea, right? So you have all those genomes. Some bacteria we don't know, we don't have their complete genome sequence, but we have all those sequence reads, and you can perform assembly to construct something called contigs. And those contigs, basically, just think about contigs as a part of the genome, but not the whole complete genome sequence, right? It's a sections of the genome form a contigs. And after you construct those contigs, you can further bin or cluster those contigs into the bins. And each of the bins of the contigs can be treated as a potential new bacteria. And some of them belong to existing bacteria also, right? So this is called the genome assembly. So our goal is try to estimate the growth rate of those guys, those assembled genomes, which may not be complete genome, means there are gaps, right? There are gaps, we don't know. We don't know the order of those contigs relative to the replication origin. So how to do that? So let me just, so basic idea is after you construct those contigs, the bins of contigs, for each of the sample, you just align your sequence reads to those contigs. And you can have a coverage of the sequence reads on each of the contigs, and you do some normalization to take into account GC difference, taking into account the length of the contigs. But eventually what you can get are those contig specific normalized reads count or coverage for each of the samples. And what you see here are the data. This is real data from 6 different samples. On the top are the 3 healthy individuals. On the bottom are the 3 patients with an inflammatory bowel disease. The x-axis are the contigs, just some arbitrary label of the contigs because we don't know their order. The y-axis are the normalized log base 2 of the coverage. And you don't see any trend, right? You don't see any trend here because the contigs are not ordered. So my question to you is, can you order those contigs? Can you figure out the order of those contigs? And indeed, this can be done easily, but without showing you how we did it. After basically all we did is perform a PCA, use the data from multiple samples, and use the first PC to order those contigs. And you replot the coverage, the log 2 of the reads coverage over the PCA ordered contigs. Now you see this monotone trend, right? You see a nice monotone trend for the samples, particularly for the patient sample. You see a nice linear trend. Okay, so this is how we order the contigs based on PCA. So basically, idea is this. So basically, you just think about if you know the true order of the contigs along the genome, then in theory we should, for the sample. So it's here this illustration. Each row represents 1 sample, and each column represents 1 contig. And the number, the size, represent the normalized coverage of that particular contig for a given sample. So if we know the true contigs sample, if we know the order, then we expect this is a monotone order for the samples with actively dividing bacteria, right? But we don't know the order. So what we because of the limitation of the assembled genomes, those columns are not, those order of the columns are not known. So what we have is the true coverage of assembled coverage, but with a permuted columns. And in addition, we have noise. So the computation question is, if I give you this matrix like this, right? I said, okay, each row is a monotone, but I don't tell you the order. How can you figure out this order? And that actually, as I mentioned, is really done by just, all you need to do is perform a PCA, treating those columns, those contigs as an observation, and each of the row as a variable. then you order the contig, the column based on the first PC. And this indeed is the key step in our algorithm, we call it dynamic, we published a couple of years ago in Nature Methods. So as I said, the key of this approach is to apply the principal component analysis to order the contigs. After you order the contigs, then you can figure out the peak coverage and the trough coverage from which you can quantify the bacterial growth rate. So this is basically our algorithm. And we, in the couple of papers that we published recently, we also studied some of the statistical properties of this procedure. So I'm not going to go to the details, but I just want to briefly mention this idea of using the PCA to recover the order of the contigs. So the basic idea is this, right? Is we have this X to be the coverage, the GC and the length adjusted log of the reads count, which is coverage of the reads for that particular contig along the genome. And total, we have N samples and we have P contigs. So each of the coverage can be written as a true coverage matrix plus some noise, some Z noise. And this true matrix will assume for each of the individual, each of the row, for each given row, this matrix, each row, you have a monotone matrix. Each row is a monotone over different columns. But we don't observe this X. What we observe is the permuted version of the X. So why is our observed context coverage without knowing the order can be treated as a permuted version of the true coverage? And the goal is, if I give you this Y, I want you to recover this permutation. And this we call this a permutation recovery problem. And indeed, it's done by performing the PCA. And we study some of the property, basically shows under certain conditions, this PCA is indeed can recover the order of the contigs. So I'll skip that. So after you figure out the order of those contigs, the next step is try to figure out the peak coverage and the trough coverage. And what you can do is you use all the sample to figure out the order of the contigs. Then for each of the sample, you fit a linear regression line. And from that fitted linear regression line, you can estimate the peak, the highest value, mean value of that regression line, and over the smallest value. So those are the peak and trough ratio. And we can use that to estimate the bacteria growth rate. So anyway, everything I did here is implemented in this software package called Dynamic, which are available now. Okay, so this is what I want to say. Let me just give you one example. I have been working with Crohn's disease in the last 5 to 10 years already. So this is one of the example where we look at the children, pediatric patients with active Crohn's disease. So 90 children, they have the active Crohn's disease, and they were treated by 2 different treatments. One is the diet, this is enterotherapy. So 38 of them were treated by enterotherapy. And 52 of the kids, of the children, they were treated by anti-TNF therapy. And those treatments were not randomly assigned. It's really determined by the treating physicians. So, and we give them treatment, and the baseline, we measure the stool microbiome by performing shotgun metagenomic sequencing. And during the follow-up, so we follow those kids for a total of 8 weeks. And so week 1, week 4, week 8, we obtain the stool microbiome samples and perform shotgun metagenomic sequencing. In addition, we also have 26 healthy normal children, and we have their gut microbiome. So for this study, we did a shotgun metagenomic sequencing. In average, we have about 11 million pair and sequencing reads. So it's a fairly large data set. So over the 8 weeks of treatment, in the anti-TNF group, about 50% of the children responded to the treatment. And in the enterotherapy group, about 32% of the kids responded to the therapy, okay? So in our original 2015 paper, we just look at how, after treatment, how the gut microbial community shift in terms of the composition, the community composition. So that was published before. But you can apply our algorithm and try to study whether there's a difference between the healthy individual and the children with Crohn's disease in terms of how fast the bacteria are divided. So our algorithm then we're able to estimate the growth rate for a total of 278 species, and about 20% of those species appear in 50 or more of the sample. Sometimes you don't observe this species in a given sample, then you cannot quantify their growth rate because you don't even observe the species, right? There's some missing data problem, but if you're missing, we just assume they don't carry that particular species for a given sample. So then what we did, we quantified the growth rate for each of the 278 species. Then we just compare whether there's a difference in the growth rate between the healthy individual and the Crohn's disease sample by using the simple two-sample test, two-sample rank-based test. And you can see here with FDR at a 5% level, we identify a total, I think, 7 of the assembled bacterial genomes that show a very big difference between the healthy individual and Crohn's disease individuals. In addition, those assembled genomes, most of them, they cannot be assigned to currently known bacterial species. So there's an algorithm you can try to annotate those assembled genomes. Now, sometimes you can only say they are bacteria. You cannot even annotate them to the order or phylum level. So most of those bacteria, those assembled bacterial species, they have higher replication rate than in the healthy individual than in the Crohn's disease patient. This is one, they have a lower growth rate, okay? And you can see the difference are quite big. For example, here I give you 3 examples, and here are their annotation. You can see a very different, the estimated peak to trough ratio between the healthy individual and Crohn's disease patients. But what is also interesting for those 3 species is after treatment, I didn't even separate the treatment into diet or anti-TNF treatment. You can see those guys, the growth rate started to go up. And eventually, after 8 weeks of treatment, they're getting closer to the normal individual. See here, this one, the patients get a low growth rate after treatment going up. This guy actually got a higher growth rate, but after treatment started to go down. So it seems like the treatment is doing something to the growth of some of the bacteria based on our analysis. So this basically provides another way to study the microbial community on the top of the standard relative abundance of the bacteria. Okay, so this is my second example. So I'll stop here and see any questions.
Yeah, we have a couple, and I think you may have just answered this one, but I'll say it nonetheless. So we were asked, could we be more interested in which bacteria differ among groups?
Yeah, which bacteria are different in terms of relative abundance, in terms of rate of replication, in terms of right, you have to fix some feature to study. Yeah. One interesting question would be try to link the rate of replication to the abundance of the bacteria, see how they are related. But there's some difficulty in linking those two, because if you know the sequencing data does not provide absolute abundance quantification. We only have relative abundance of the bacteria. So we didn't try to link those two type of features. Yeah.
And then I have a question too. So in your previous part, in part 1, you had mentioned the same Crohn's disease, and you compared healthy, healthy, I guess, patients or people with those with inactive Crohn's. Did you see that? So in that slide, you showed that the healthy patients grouped more closely together. They were more similar, and in the Crohn's disease, they were like much more variable, had much greater distance. Did you get a chance to compare patients who were trying to control their disease with diet versus with anti-TNF treatment? And did you see whether that impacted the sort of spread?
That's a good question. Good question. So this diet, first of all, this diet treatment data is for the gut microbiome. The previous one is the tissue, the colon tissue microbiome. So, but this one is good. So, yes, we did in the paper. We did look at the distance to the normal centroid after treatment. So after treatment, those patients, their microbial community in terms of distance, indeed getting closer to the normal centroid. Yes, we look at that.
Thank you. Yeah.
So so Nadia, I have another 30 minutes, 20 minutes.
Yeah, however long. We've had a lot of people have ended early, so if you can, if you have extra slides and you want to keep speaking, yeah, we're happy to have you have another 30 minutes.
Okay.
But if not, we
do
can
have
also
the last
Example.
example.
Perfect. Perfect.
It shouldn't be too long because Ming told me about 1.5 hours.
Yes, absolutely. So you still have half an hour. Yeah.
Okay. Great. Okay. So if no further questions, I'll just move on to the third example, which is more computational example. So here's example 3. So as I mentioned, one way that microbial community can affect human health is through producing many really important small molecules and the secondary metabolites. Really important. Some, for example, some of the antibiotics, known anti-cancer reagents, they were actually produced by those microorganisms. So those secondary metabolites, usually, if you look at a bacterial species, you want to see whether they can produce certain small molecules or secondary metabolites. You actually need to look at the genes carried by those microorganisms. So those secondary metabolites are encoded by the genes and tend to cluster together in the genetic package. And this cluster of genes altogether is referred to as a biosynthetic gene cluster. Okay, so this is how bacteria produce certain small molecules if their genome carries a certain cluster of genes. So here I just give you a couple examples. So what you see here, those are the 4 different biosynthetic gene clusters, known biosynthetic gene clusters. They belong to different bacteria. For example, the first one is E. coli, and they can produce different or similar secondary metabolites, right? Small molecules. So this is the one BGC. Here's another third BGC. You can see they're different, right? They are not identical. They're different in terms of number of genes. They are different in terms of length of the genes. However, if you look at the function, so each color here represents the genes, the function of those genes, certain function. For example, the black color represents the genes with a function transport and other function, including ACP, dehydratase function, different function. So just look across the different BGCs, although their genome sequence may be different, but they do have some similar function. So the question is, now we have a database actually that include about 2,000 this type of BGCs from different bacterial genomes. And the question is, can we identify other BGCs based on the sequencing data? Can we identify all the BGCs based on the sequencing data? So this was initially studied by a 2014 paper published in Cell, and they published an algorithm called the cluster finder, which is basic standard two-state hidden Markov model-based approach to identify other BGCs in the genome. So here is what I mentioned. There's a database called MIBIG database, which is a repository of the currently known BGCs. And there are 2,000 some known BGCs. So go to this database, you can see those are the BGCs and tell you what are the genes involved. And they also tell you some information about description of the BGCs and which class. Sorry, my dog is barking. So you can classify those BGCs into different biosynthetic class. For example, this one is a polypeptide BGCs. And you can see which species carry this BGC. So you have all those information from this database, okay? So as I said, our goal is this, is now we have about 6,000 bacterial genome sequence, complete genome sequence of about 6,000 bacteria. And our goal is try to identify and characterize all the BGCs. Characterize means the BGCs can be classified into different biosynthetic classes. We want to see which class they belong to. And that's our goal, okay? So we treated this problem as a supervised learning problem because we have training, right? We know 2,000 some BGCs. We know their gene sequence from the database. So what we did is for those 2,000 BGC gene sequence, we convert those DNA sequence into apply gene prediction to the sequence to predict the gene, to predict the open reading frame of gene prediction. And from the predicted gene, we further convert the data into a sequence of protein family domains. Those are the different protein family domains with the different names. So altogether, based on those known BGCs, we have about 3,685 protein family domains. And those BGCs belong to about 1,094 bacterial species. So those are the training, right? Those are the known BGC information. Then in addition, in order to perform prediction, we also need negative controls, right? We need non-BGCs. So what we did was, this essentially follows the original Cell paper. We look at those reference genomes, 6,000 some reference genome, and we convert their genome sequence also into the protein family domain sequence. So total is 11,000 unique protein family domain names. And from which we control, it's just basically randomly pick some of the domain names and put them together to construct the control, the non-BGCs. So you can talk about 10,000 controls, about 10,000 controls, 1 to 5, about 1 to 5 ratio. Okay, so this is how we get the training data set. And the actual algorithm we used is called the recurrent neural network, so-called long short-term memory recurrent neural network. It's one type of deep learning algorithm that works pretty well when you have this sequence type of data, right? So you convert the data into a sequence of protein family domains. Those are basically a sequence of different names, different words. So in order to run this deep learning algorithm, we need to embed all the information we know about those BGCs, including the protein family domain names, protein family clans. Basically, we can group the protein family domain into different groups with similar function, similar structure. And also some description of the protein family function information. So 3 types of information, and you can embed those information by using some numerical embedding, such as word-to-vec embedding. So let me just give you some idea about those BGCs. So those are the known BGCs that include a minimum of 3 PFAM domains. So here are the histogram, the count of the BGC. So basically this just give you basically saying most of those BGCs, they only have a fewer number of protein family domains, like only a few words. It's a shorter sentence, think that way. And a few of them, they are longer. For example, a few of the BGCs, they can include, for example, 20-some protein family domains. So it varies. The length of the BGC in terms of the number of protein family domains varies from 3 to about 250, okay? Different size, okay. So as I mentioned, we also use a protein family clans information. So this information basically that you can group the protein family domains into different clans based on their sequence structure, function similarity, and the similarity between the profile hidden Markov model. So basically this gives us some information about how similar two protein family domains are in terms of function and structure. Okay, second type of information we need to use. And the last information, which I didn't talk about, is so each of the PFAM, if you go to the database, there's some description, function description of that particular protein family domains. So we can also use those information into our embedding. Okay, so eventually what we did is we treated each of the BGC as a length of 256 protein family domain names. For the shorter BGC, we just use a 0 padding, make them the same length. And for each of the PFAM, we use the numerical embedding, use a vector of 1,126-dimensional vector to embed the information about domain name, PFAM clans, and the PFAM function description information into that. So basically each protein family domain name now is represented by a vector of 1,126-dimensional vector to code that way. Then you can just put into the standard LSTM recurrent neural network model to build the prediction. Okay. So the other really interesting idea to use is the so-called data augmentation ideas. This idea provides a way of potentially increase the prediction performance when the sample size is small. So the basic idea is, for example, if I have a BGC, and remember my BGC is represented by a sequence of protein family names. So during this deep learning steps, each of the epoch, we can slightly perturb the original data. So that's called the data augmentation. Basically, you can shift, you can change one of the protein family domain with a similar class, in a similar class, similar function. You just replace a protein family name randomly with certain probability, replaced with another protein family name. And this is called data augmentation. I'll show you this actually indeed help us improve the prediction performance. So here's a plot. I want to show you a couple of plots now. Okay, so what you see here are each dot represent one known BGC. So each dot represent BGC. And remember, those BGCs can be classified into different functional group. So polypeptide, RIPP, saccharide, terpene. So those are the different BGC groups, and they are colored by different color. So by different color. And what you see here is a t-SNE plot of the training. This is for the training data set. So when you run the deep learning, prior to your outcome, the class label, you have all those vectors. And this is a t-SNE plot based on those vectors. And you can see the BGCs of different types. They indeed separated pretty well, right? So this different color represent different types. Decent, well, but we should expect it separate well, because this is based on our training data set, training set, okay? Except there's a red color, there's a yellow color, you see them spread out a little bit into different class. The yellow color are the BGCs that belong to other class, other unknown class, or sometimes they have multiple function class. They belong to multiple function class, okay? So this is for the training. This basically tells us that the embedding seems to work, right? The numerical embedding can indeed capture the difference of the BGC for the training data set. Now, how about in the testing data set? So testing data set means we actually leave some of the bacteria species out. We never use them. Then we apply our algorithm to the testing data set. This is similar type of t-SNE plot for the BGCs in the testing data set. Those are the red, and all those are non-BGCs, a lot of non-BGCs. And you can see they are separated well. Means this algorithm can indeed separate the true BGCs from non-BGCs, right? It's in the testing data set. So this table just gives you a little more detail of the comparison of different algorithms in terms of predicting different things. So I just want to point out those different algorithms. Deep BGC, this algorithm published in 2019, so one of the, I think the first deep learning algorithm that was used for predicting the BGC. And our research was really motivated by this paper. And deep BGC class, this is try to predict the BGC class. Deep BIG class. This is our algorithm. Deep BIG, this is the algorithm that only predict BGC, non-BGC. In other words, deep BIG make a binary prediction. Deep BIG -O, this is the algorithm that we do not use the data augmentation. It's basically, I just want to show you this data augmentation can actually help us make a better prediction. So if you look at this column, the last column, and this column, you can see in terms of the performance of the prediction in the testing data set, and you can see the data augmentation really helps. So let me just explain a little bit more about what is a testing data set. So there are 9 genome, bacterial genome, total 13 contexts, and there are, from the database, there are 290 known BGCs. And those BGCs, we never use them in the training. This is known, right? For those, those are known BGCs, but we know they may have other true BGCs in there. So we have to create artificial genomes, make sure they don't have other BGCs. Otherwise, how are you going to evaluate your prediction performance? So we basically create 130 artificial genomes with known BGCs, and the other part of genome, they are replaced by non-BGCs. Okay, the BGC with non-BGCs. And now you give me this artificial genome, then I can make a prediction. Okay, so evaluation is done by both at the PFAM level. So basically, you give me a protein family domain, you tell me whether this belongs to BGC or no BGC. And so that's an initial output from the deep learning algorithm. Then you have to do some post-processing of the protein family domain level prediction into a segment, because we want to see a cluster of BGCs, right? A cluster of protein family domains. And this is a PFAM domain BGC. So this is truly the biosynthetic gene cluster after post-processing. So we predict a segment of the genome, right? Now, how are you going to evaluate how real those predictions are? So you basically what we did is to compare the predicted BGCs with the true BGCs and see how much they overlap. If they overlap by 20%, 40%, or 100%. If it's overlap 20%, you make a right prediction, and this is the result. If they overlap by complete overlap between the predicted BGC and the true BGC, and this is the performance. And what you see here, the numbers are the F1 score. And you just compare our algorithm for binary prediction, our algorithm, and the original deep BGC, you see a nice improvement over original BGC. So what we did differently from the original deep learning algorithm is, I said, we incorporate some additional information such as function information, similarity information of the PFAM domains. So that additional information truly help us to make a better prediction. In addition, this data augmentation step also improve from our prediction. Okay, so this is for binary prediction. We also evaluated the prediction for the class, BGC class. And so what we did is we built our algorithm based on this database version 1.5, 1.4. And the newer version of so the database keep getting updated. The newer version of the database, people deposit another 160 new BGCs. So we just say how well our algorithm can predict those 160 new BGCs, okay? And so here is a similar type of t-SNE plot, but it's for the new BGCs. And you can see they are, okay, they're separated, somewhat separated, but not as clear. And one of the reasons is for some of the class, the training sample size is fairly small, so make it quite difficult to predict because the sample size is small. You see some separation in terms of different type of BGCs. And this plot, this is a similar plot. This is a predicted BGC. And what you see here, the BGC can be summarized at the protein family domain level or the gene level. And we'll see here, last column is the deep BGC algorithm. This is our algorithm. This is the deep BGC, but you make a multi-class prediction. Okay, so you would just compare again our algorithm with the deep BGC, you see a fairly decent improvement in the prediction performance. It's exactly the same data set that we used here. Okay, so then what we did is we applied our algorithm, this prediction algorithm, which is to all the bacterial genome, to all the 5,666 bacterial genome, and then see how many BGCs we can predict. So eventually we predict 170,000 predicted BGCs. It seems like a lot of BGCs, but this is actually still smaller than some of the BGCs predicted in terms of number by the two-state hidden Markov model. So in average, each of the bacterial genome in average, they carries about 30 BGCs, and the number of BGCs range from 0 to 243 BGCs. And here, just give you some idea about those predicted biosynthetic gene clusters. They, just in terms of their length, relative to the length of the genome, you see they covered about, mostly covers about 10% to 20% of the genome. Even we predict this many BGCs, they still a small part of the bacterial genome here. And this C plot, this C basically to see for each of the genome how many BGCs. I already mentioned the average is about 30. For some of the genome, they have more than, they have like 243. And some genome you have 0. We didn't make any prediction for some of the genome. Okay, this one, the last plot is a histogram in terms of how many genes. You see, most of the predicted BGC, they have rather smaller number of genes, like 3 or 4 genes. The first plot will basically just show you the percentage of the BGCs that we predicted based on our algorithm. So this is a deep BIG algorithm as compared to the training data set. In the database, in the MIBIG database, what are the proportion of the BGCs in different categories? And our predicted is not quite the same as what we have in the training data set. There's a one class of the BGC, which is the RIPP. You see, we make a lot of predictions. And the reason our algorithm make a lot of prediction is this class of BGC, they are very much conserved. All the BGCs belong to this RIPP class. They are very similar, so make it very easy to identify them. And this is why we have a bigger percentage of predicted BGC that belong to this RIPP class. Okay, so this is all I have is I'm still with so we are still thinking about how to convince people, how to validate those computationally predicted BGCs. There's a lot. So we're still thinking about some ways of trying to convince ourselves that those predicted BGC, at least most of them are truly the true BGCs. So this is still something ongoing there. Okay, so this is also my first attempt of applying the deep learning algorithm to this type of problem. I think it's in terms of predictions performs extremely well as compared to the standard approach such as two-state hidden Markov model approach, because of the algorithm is able to capture potential long-term and bidirectional dependency of the appearance of certain protein family domain. Okay. So here, just to summarize what I presented, I just briefly show you 3 problems and the statistical method that or computational method that we developed it in analyzing the microbiome shotgun metagenomic sequencing data. I show you some good empirical performance, and for some of the method, we do have some theoretical understanding of our algorithm. So in terms of the next steps, my group and my collaborators now, they are very interested in understanding how the microbial community, the role of microbial community in precision nutrition, microbiome, and health. And also here, my collaborators are really interested in understanding the nutrition and the host immunology through the studying of microbial community. So with that, I'd like to thank my collaborators, all my students and postdocs that work with me on various projects, and my longtime biology collaborators, Gary Wu, Rick Buchman, Jim Lewis, Kyle Bittinger, and the people in their lab. And also Tony Kai, my long-term statistics collaborators. So that's all. Thank you very much.
Thank you so much.
Question?
Really, really wonderful talk. And I have another question. So it was a really great idea to incorporate the additional information into DeepBig, and it showed pretty amazing improvements in prediction by your method. I was curious, in the training data set, you mentioned to be able to use it on smaller sample sizes where you replaced protein domains by other domains with similar function. How did you handle the domains that you described as other, that you said sometimes even had like 2 different functionalities associated with them? So did you, I guess, take it into did you look at more granular level details to be able to substitute some of the other domains for I guess, for others, or did you just not not try to substitute, one other domain for a different one? I don't know if I'm being clear.
It's a great question. I know what you're asking. So the two things. One is the BGC class. The sum of the BGC they may belong to different class. Those are the prediction part. And so if they belong to different classes, we couldn't predict them. We just assign them as other. But here, the similarity is at the PFAM domain similarity level. So there's a database called the PFAM clan database. And that actually tells you a set of PFAM that are similar. So it's almost like tell you some of the words, they have similar meaning, right? So original deep BGC, they only use a word. We use the word with similar meaning. And also, we use a description of the word to tell the machine there's some function, description. So we also use that. So those are the three important improvements over what they did, what they published. Yeah. So that's why I want to show you, for different improvement, whether we can gain anything. And I think this plot basically to show you with imputation, without all the rest are the same. This one's with that augmentation. This is without augmentation, we can see improvement. Yeah.
Thank you so much.
Sure. Any other questions I can answer? Good. And yeah, I think you are monitoring the chat.
Yes, yes.
Okay.
Some of those that I asked were in the chat.
Okay. All right.
One question is asking whether you can share your slides.
Ask you what I mean. But this is recorded.
Yeah, I think we'll post the video and also the slides after that.
Okay.
Okay, yeah, thank you so much for great talk. And yeah.
Good seeing you. I also see at least Dabao's picture. So I don't know whether he's there. So, yeah, great seeing you guys. Have a great summer.
Yeah.
Yeah. All right. Good. I'll log out.
Okay.